Hello, everyone, and welcome to chapter 14 and 15. This will be the first part. We're going to be focusing mostly on neuroanatomy, specifically the brain's anatomy, and looking at some things there that I want to talk about. So, I chose the quote: hardships often prepare ordinary people for an extraordinary destiny. And this class is certainly a hardship. So, there you go. This is what I'll be testing on for sure, things like that. So let me go into, I tell you this, I always list some of the organs that we talk about, some of their basic parameters, anatomical parameters. I never test on like average weight, average volume of a brain, but brain, I will say this, is 97% of your nervous tissue is in the brain. It's roughly about 1.4 kilograms. That's about 3 pounds. It's typically about 1,200 milliliters in volume, but it can be as small as 750 in a normal functioning adult, as high as 2,100 milliliters in a normal functioning adult. Um, Brains of males are about 10% larger than females, but there is no correlation between brain size and intelligence. What this really has to do with more than anything is males are on average bigger. Remember, there are four regions of the brain. There's a cerebellum or cerebrum, diencephalon, and brainstem. I don't know why I said cerebellum first, but cerebellum, diencephalon, and brainstem. Now, when we start this, let's start with the cerebrum, our first major region, and that is the cerebrum. It's by far the largest region. This is where we do all of our thinking. This is where our higher order functions, our thought, our intellectual functions. What I like to say, the higher order functions. On them, there are gyri. That is a singular gyrus. These are the elevated ridges that look like folds that stick out. That is to increase the surface area of the cerebral cortex. There is the sulci, and those are the shallow depressions, and they give us the little recesses. There are fissures. These are deeper. Sorry, I keep yawning. I'm very tired. I apologize. I don't want to restart my recording. There is gray matter. This is found in the cerebral cortex and the basal nuclei. There's white matter. It's under the cerebral cortex and around the basal nuclei. There are your hemispheres. There's a left one and a right one. And there's a longitudinal fissure that separates the two hemispheres. All right, the cerebellum is your next. It's our second smallest part. It's our little brain. It's the second largest. It's one of the things it does is it takes what I actually intend to do versus what I actually do. What I intend, the motion I intend to make, the motion I did make, compares those movement patterns so I can make the same motion repeatedly. It also helps regulate posture and balance. Our diencephalon is located actually between our cerebral hemispheres. It's superior to the brainstem. So here we have our brainstem in the green, blue, and reddish color, orangish color. This more reddish color right here is our diencephalon. The diencephalon has our thalamus, the region called the hypothalamus below that, and the pituitary gland. Our thalamus is a relay center for every bit of the senses except for smell. It sends it to the right part of the brain with the exception of smell. The hypothalamus actually forms the floor of the diencephalon, and it's an endocrine structure as well. It contains the centers involved in autonomic functions. Very important. We'll talk about that in AP2. The pituitary gland, it's an endocrine gland that releases 9 hormones with the infundibulum, that is the stalk that holds the pituitary gland. This is integrates with the nervous system with the endocrine system. The brainstem, this is made up of our medulla oblongata, pons, and midbrain. Our diencephalon, this is a structural thing that connects between our cerebral hemispheres to our brainstem. Our brainstem has a bunch of centers that are important in processing things called nuclei. Nuclei is a collection of neuron cell bodies in the central nervous system, and they relay information that's coming and going from the cerebrum and cerebellum. Okay. The midbrain, the mesencephalon, this is the short section between our diencephalon and the rest of our brainstem. This is called the midbrain, part of the brainstem. Right here is our midbrain in green. It maintains consciousness and does subconscious motor controls. Our pons is going to be our bridge between our cerebellum and our rest of our brain. It relays between the medulla oblongata and the cerebrum, connects our cerebellum to our brainstem, so it is the bridge. It is contains visceral motor nuclei and somatic motor nuclei, both, and there are two respiratory nuclei in it. The medulla oblongata, which is right here in this peachy color, this is the inferior part of your brainstem. It's continuous to the top of the spinal cord, superior part of the spinal cord. It has all the sensory and motor tracts that communicate between the spinal cord and the brain, and a lot of vital reflex centers like the cardiac control, respiratory rhythmicity, vasomotor, vomiting centers, and things like that. Okay. All right. Now, let's talk a little bit about how we got our nervous system. The embryonic development determines basically the organization of our brain. Embryonic development. Now, there is what happens in our CNS starts from what's called a neural tube. At 3 weeks, the neural tube basically swells and enlarges into three what is called primary brain vesicles: a proencephalon called the forebrain, a mesencephalon called the midbrain, and a rhombencephalon called the hindbrain. The proencephalon becomes your telencephalon and diencephalon, and we'll see what the rest of this stuff becomes. Then, after 6 weeks, the previous three develop. 6 weeks, previous three develop into these at 6 weeks. The telencephalon forms what's called the cerebrum. The diencephalon forms your thalamus, hypothalamus, and pituitary. The mesencephalon forms your midbrain, and that's got the visual and auditory sensory centers and centers that maintain consciousness. Your metencephalon's got your cerebellum and pons, and your myencephalon forms your medulla oblongata. The medulla oblongata, pons, and membrane are all combining to make our brainstem. Do know what the cephalonic divisions become. That will be on the test. Okay, so our forebrain, midbrain, hindbrain swell, and they become our telencephalon, diencephalon, mesencephalon, metencephalon, and myencephalon. Now, inside the brain, there are ventricles. These are chambers that contain cerebrospinal fluid, or CSF. There are two lateral ventricles, the left one and a right one, a third ventricle, and the fourth ventricle. Separating the two lateral ventricles from each other is the septum pellucidum. Connecting the two lateral ventricles to third ventricles, interventricular foramen. The cerebral aqueduct, mesencephalic aqueduct, or aqueduct of midbrain, that connects your third ventricle to your fourth ventricle. And inside these structures are choroid plexuses. And what these are is capillaries and ependymal cells that come together to produce and maintain cerebrospinal fluid, or CSF. Okay, so here are your ventricles. Okay, now we said there is cerebral cortex and the basal nuclei that are involved. Okay, now the meninges. The meninges, they surround your brain and spinal cord. There is the dura mater, and the dura mater is tough, white, dense, and fibrous. It is your outer layer connective tissue. It's got an endosteal layer right here, and which is sometimes called periosteal layer, and it's got a meningeal layer right here. Then underneath that, there is a subdural space. Then our arachnoid mater, and our arachnoid mater has these little things that look like spider webs in it called arachnoid trabeculae. The arachnoid trabeculae. So this is the middle layer that covers your blood vessels. Then we have our pia mater. Okay, our dura mater is tough, white, and fibrous with the endosteal layer and the meningeal layer in a subdural space that is below, separates the dura from arachnoid. Then the arachnoid has the arachnoid trabeculae in it, which is delicate collagen and white fibers. Then there is in that space right here is called the subarachnoid space, and then there's a pia. That is a meshwork of elastic and collagen fibers that surround blood vessels and firmly bound to the underlying neural tissue. You will see it on the spinal cord as well. Now, in the brain, there are dural folds. The dural folds are they are associated to the dura that are folded around the brain. The word falx means sickle. So there is the falx cerebri that's located between the two cerebral hemispheres in the longitudinal fissure. There is a tentorium, sometimes called the tentorium cerebelli, and a falx cerebelli. Falx means sickle. Cerebellum and cerebrum is separated by the tentorium and the falx cerebelli. I just learned it as tentorium when I was a student. Cerebrospinal fluid, CSF, cushions your neural tissue, supports the brain, transports nutrients, chemical messengers, and waste products. That's what it does. It is kind of like a blood-like fluid that does the transport. It surrounds and bathes our neural tissues of our CNS. Now, CSF is produced by capillaries and epidermal cells. These are called the choroid plexuses. You will find them in all four ventricles, except they will not be in the aqueduct and midbrain because that would strangulate it. And there's parts of the lateral ventricle that it's missing in. The CSF produced by the epidermal cells or choroid plexuses secretes in the ventricles. Then basically as it secretes in the ventricles, we secrete it into the ventricles. For example, here, third ventricle. What's going to happen, it gets into the choroid, from the choroid plexuses to the ventricles, then goes in the central canal, passes out and around the spinal cord. Okay. Now, CSF is produced by the epidermal cells, but it is absorbed by arachnoid villi and back to venous drainage as a waste product. There is a blood-brain barrier to prevent materials that are in the CSF, things that are in the brain, or in the blood, rather. There we go. Let me get this right. Isolate the blood from the CSF so that we keep things like pathogens that's in our blood away from our CSF. The ependymal cells, they have gap junctions, and this acts as a filter. And there's small amount of liquids and gases that can pass through. This keeps large molecules in. Lipid soluble things like, I really need to put the CO2 as a subscript and the O2 as a subscript. I apologize, I never did do that. But it keeps things like CO2, O2, lipid, steroids, and alcohol. They can pass through the lipid membranes and into the CSF if they can pass through. But things like sodium, hydrogen ions, potassium, and chloride, and some neurotransmitters, they can pass, but only highly regulated. They have to be very regulated. They have to go through proteins and things like that. CSF is found in the brain between the arachnoid and pia mater in the subarachnoid space. There is a blood CSF barrier as well. That's actually epidermal cells that connect by tight junctions. They actually surround capillaries in the choroid plexuses, and they limit things getting into the CSF. But composition of blood and CSF can be very different from each other. Okay. Now, blood-brain barrier is lacking in four places: the hypothalamus, the posterior pituitary, and the pineal. Why? These allow hormones to pass through the bloodstream, all endocrine things. They are also lacking in the place that the CSF is produced, the choroid plexuses, because if we had a blood-brain barrier here, we never would allow CSF to actually be produced. Sorry if you hear my dogs barking. And they want something. They've been fed, they've been out, but they want pets and they want my attention. I've had to be gone today. The medulla oblongata is a relay thing between the brain and spinal cord. There's reflex center, there's cardiac control, respiratory rhythmicity, which is breathing rate, vasomotor, which is blood vessel diameter, and vomiting centers that are all found in the medulla oblongata. The brainstem has our sensory and motor nuclei in the pons for cranial nerves. They have nuclei involved in respiration. One's called the apnoestic center, one's called the pneumataxic center. We will talk about these in A&P2. The nuclei on tracks that process relay information sent to or from the cerebellum. Okay, as well. Now, the corpora quadrigemina, one of my favorite structures to talk about. Here we have the two. We have our corpora quadrigemina. We have our two superior colliculi and our two inferior colliculi. This, they control the turning of the eyes and head When noises, they compose four rounded elevations called colliculi. There's two superior. They serve as the reflex centers for the eye and head movements in response to visual stimuli, like if somebody walks by you. And inferior colliculi, they serve as reflex centers for head and trunk in response to auditory stimuli, like a loud car backfires or a gunshot goes off or somebody yells boo or something like that. Okay. The cerebellum is reflexive, learn patterns, muscular activity subconsciously. There is structures on it. There's a vermis. It means worm-shaped. It divides us into two hemispheres. There is a left and right hemisphere. There is a primary fissure that separates it into an anterior lobe and a posterior lobe. There's the folia that look like the folds of a book. Inside, there's the cerebellar nucleus, and there's all the arbor vitae. Okay. The cerebellum does the adjusting of postural muscles. It creates a rapid automatic adjustment to maintain equilibrium or balance. It modifies activities and motor centers of the brainstem so we can adjust how our things move. We also program and do fine-tuning of conscious movements like riding a bike or playing a piano, controlling things like that. One of the things we do is refine learn movement patterns. We take what we intend to do and what we did do and combine them together and adjust our movement. It also compares motor commands with proprioception, which is body position, and adjusts our muscles to help produce movements that are smooth. Okay. All right. The diencephalon, the thalamus and hypothalamus. The roof is the epithalamus, roof of the diencephalon. Associated here is our pineal gland. Pineal gland is the endocrine structure that makes the hormone melatonin, which is involved in what we call our circadian rhythms, maintains our daily cycles, and our yearly circadian rhythms, our biological clock. Our thalamus is a large oval-shaped structure. It's 80% of your diencephalon. It is like the old-timey telephone operator who name a number, please, and plugs you into the right place. It takes sensory information with exception of smell and sends it the right part of the brain, to the right part of the parietal lobe. The hypothalamus is the floor of the diencephalon, just above the optic chiasm. The thalamus filters, relays information from the spinal cord and cranial nerves go to the right part of the cerebral cortex. It takes information between the basal nuclei and the cerebral cortex. Very important is the basal nuclei. That's dysfunctions with that cause cerebral palsy. And the third ventricle separates your left and right sides. Okay. What does the hypothalamus do? A lot of things. It makes ADH and oxytocin, which are hormones. It regulates your body temperature. It controls autonomic functions. It does voluntary and autonomic function coordination as well. It coordinates nervous system and endocrine system. It regulates circadian rhythms, your day-night cycle. It actually does subconscious control skeletal muscles. And it does all your motions and behavioral drives. And I said it really is involved in your 4 Fs, feed, flight, fight, and mate. It has a feeding center, your hunger center, your thirst center, thirsty, and your satiation center, what we call satiety, and that means I feel full. Okay, regulates food intake that I feel full. Okay, the limbic system is something important I talk about. Very important, establishing emotional states, conscious function between cerebral cortex and autonomic functions, and facilitates memory and storage retrieval. Very important is the limbic system for emotional memory. It's a big thing in the impulsivity and stuff like that, memory issues of people with ADHD. The cerebrum, our central sulcus, separates our central sulcus right here, separates our frontal lobe from our parietal lobe. There is a precentral gyrus on the frontal lobe and a postcentral gyrus on the parietal lobe. The precentral gyrus is the primary motor cortex, and the postcentral gyrus is the primary somatosensory cortex. There is a lateral sulcus, which you can spread apart and see the insula between there. The parieto-occipital sulcus separates the parietal and occipital. Inside, there's a corpus callosum, is a thick band of white matter connects your left and right halves. There's a cingulate gyrus, which is part of the right above the corpus callosum, and the olfactory bulb and tract is on the bottom of the frontal lobe, and it carries smell. We can't see all of that, but amygdala is a thing I want to talk about more. There's some areas I want to talk about a lot more. I really want to talk about the amygdala and the hippocampus a little bit more. Now, when we talk about the brain, we'll talk about principles of the brain. We are getting close to being done. We'll talk about the cerebral hemispheres, receive somatosensory information from and send commands the opposite side of the body. That's very important. What happens is cerebral cortex receives information if the right half of the brain receives information from the left half of the body, and the left half of the brain receives information from the right half of the body. One of the other big things that is important is it also the left half of the brain controls the right half of the body, and the right half of the brain controls the left half of the body. The other thing is the correspondence between what the brain does and the specific area that does it is a very imprecise definition. It's not the only place on the brain that does those things that we're going to talk about. Now, there are some things we want to talk about. The central sulcus separates your primary motor cortex and your primary sensory cortex. The motor areas, the primary motor cortex, is on the precentral gyrus. The primary somatosensory cortex is on the postcentral gyrus. The frontal lobe, to remember the lobes of the cerebral cortex, remember Freud tore pants off. Frontal lobe is going to be voluntary control of skeletal muscles, cognition, reasoning, and logic. The parietal lobe is the primary sensory cortex, conscious perception of touch, pressure, vibration, pain, temperature, and taste. Occipital lobe is our visual cortex, does conscious perception of visual stimuli. And the temporal lobe is our auditory cortex and olfactory cortex that functions to actually consciously perceive auditory and smell. Okay, hearing and smell. Definitely know these. These will definitely be on the test. Okay, I will tell you some things that always are on my test. Okay. Now, I like to talk about different fibers in the brain. There's association fibers. They are connection within one hemisphere, so they are within an association between the left hemisphere connection. That's association fibers. Right hemisphere association fibers, okay? They're within the same hemisphere. Commissural fibers, bands that connect between left and right halves of the brain, between the two hemispheres. A great example, this is the corpus callosum, is a really big place we find commissural fibers. There's another place we find commissural fibers that is in the massa intermedia or intermediate mass, or sometimes called the interthalamic adhesion. The projection fibers, they are going to go up and down. They are going to go from they basically link the cerebral cortex to the diencephalon, cerebellum, and the rest of the spinal cord. They pass through the diencephalon. This is all the ascending and descending tracts of the brain are projection fibers. Everything coming up and down to and from brain. The basal nuclei are masses of gray matter within the white matter. They direct subconscious activities. We do know dysfunction of them are linked to cerebral palsy. We do know we have special sensory cortices, that we have visual cortex. It's on the occipital lobe that receives visual information. The auditory cortex on the temporal lobe. The olfactory cortex on the temporal lobe. And the gustatory cortex is associated to the insula and on the frontal lobe itself, receiving information from taste. Association areas that we have, they are connecting sensory and motor regions of the brain. They interpret incoming data and coordinate a motor response. There's a premotor cortex called the somatic motor association area, coordinates your own movements. That's an area that's important, is your premotor cortex is right here. Prefrontal cortex doesn't fully develop till you're about 25. Now, some clinical areas of the brain that I like to talk about, some integrative centers, specifically Wernicke's, Broca's, and prefrontal cortex. The integrative centers, these are in lobes of the cerebral cortex. They receive information from association areas. They direct complex behaviors such as speech, talking, things like that. Wernicke's is language comprehension, me understanding language. It's associated mostly with the left cerebral hemisphere. It's called Wernicke's, so you get Wernicke's aphasia. That is the word salad aphasia, where we just say words that do not go together. They receive information from sensory association areas, and they coordinate access to visual and auditory memories. This is why when you're trying to think of words, you look up and things like that. And auditory, you have to listen to be able to speak these words. Broca's is our motor speech area, to actually speech production, to talk. These are primarily associated with left cerebral hemisphere as well, and they relate patterns of breathing and vocalization to talk. Prefrontal cortex on the frontal lobe, right here in blue. This area is coordinating information from all the cortical association areas. This does our very abstract things, like, for example, predicting the consequences of my actions, like driving over 100 miles an hour in a car on the interstate might be a bad thing. I might not only get a speeding ticket, but I might wreck and hurt somebody. All right. Brainwaves, I want to talk about those, and this is where we're going to stop. This will be our last thing here. Brainwaves, they can be measured with an electroencephalogram or an EEG. They are placed on your brain or skull. They can place them on the skull and the skin on your scalp. The electrical activities or brainwaves are observed, and they're measured by changing the activities in neurons per second. There's alpha waves, beta waves, theta waves, and delta waves. The alpha waves are in what we call awake adults. You're awake, but you have your eyes closed and you're resting, kind of like I'm meditating a little bit. They disappear during sleep or when you start to concentrate on a task. Beta waves, you beta pay attention. So alpha waves, awake adult. Beta waves, you beta pay attention. They go during when you pay attention. They replace the alpha waves. They are typical of individuals who are trying to concentrate on tasks under a stress or physiological tension as well. Theta, they come from time to time. Theta are transient. They during sleep in normal adults, they are common. Okay. They may appear from time to time during sleep in a normal adult, but they are very common to children and frustrated adults. Frustrated adults act like children. Theta are transient. They appear time to time during sleep. Now, delta is seen during deep sleep, all ages. They are also seen in infants who have developing brains, delta developing brains, or an adult with brain damage. Delta, deep sleep, developing brains, and damaged brains. Alpha, awake adult, beta, pay attention, theta, transient, delta, deep sleep, damage, developing brain. Okay? All right. This is where we're going to stop, and we'll pick up in the next one, guys. I'll see you in the next one. Thank you.